The overall goal of this experiment is to drive enzymatic reactions using visible light. In this approach, light is used to convert fatty acids to terminal alkenes under mild reaction conditions.
Decarboxylation requires the breaking of C-C bonds, and this bond is very stable, which makes this a very difficult reaction. The use of light is a sustainable approach to achieve such a difficult reaction.
We first had the idea for this method when we tried to add hydrogen peroxide directly to the reaction, which led to an inactivation of the enzymes. The main advantage of this technique is that it supplies a steady amount of hydrogen peroxide.
First, we use purified OLT for our light-driven biocatalysis to characterize our enzyme. Later, we also tested crude extract, which would be important for the development of industrial applications.
After cloning the synthetic OLT gene into the expression vector and transforming the plasmid into E. coli as described in the text protocol, inoculate the cells in 2 test tubes containing 3 milliliters of LB medium with 100 micrograms per milliliter of ampicillin. Incubate the pre-cultures in a shaker for 15 hours. Dilute 2 milliliters of the culture into 200 milliliters of LB with ampicillin in 2 1-liter flasks. Incubate the flasks in a shaker at 37 degrees Celsius and 180 RPM until the cultures reach an optical density at 600 nanometers between 0.6 and 0.8. Next, add 0.5 millimolar of delta-aminolevulinic acid to the cultures.